So yes, thank you all for staying for the last talk of the session. So I am a faculty member originally from NYU, New York, but I was one of the people who came over here to this unknown country, this new land, and started the CGSB Center here. And I work with a longtime collaborator, Fabio Piano, who's here. And in New York, what we've been doing for a very long time is studying basic biology using C. elegans as a model for animal development. And we thought we'd try something new when we came here, and so we thought we could use our experience in basic biology and all the molecular tools available to us to identify novel compounds that can kill parasitic nematodes. So why is this important? Parasitic nematodes actually infect over 1.5 billion people in the world, and they can cause all kinds of developmental problems and sap your energy. And they also cause a lot of monetary damage to both livestock as well as crops around the world. So it's a very big problem, and there's a small number of anthelmintic drugs that are available, and resistance is rising across the world, the same as with antibiotics due to overuse and a lack of other options. So we think it's a pretty important problem to try to develop new anthelmintics. And so that's what we've been pursuing for the last several years here. Only one problem with these parasites is that they have a very complex life cycle. So you can see here in the human, we have they migrate throughout the body to different sites in the body, and then they shed, and they're shed in excretions in feces, and then they infect another host, and then they reinfect humans. They get released actually as free-swimming organisms in the environment. And the same is true for species that infect ruminants. They shed the eggs and the feces, and then these can grow and be then consumed by other animals that are herbivores. So this is a general pattern with these kinds of organisms. So instead of trying to bring these models directly to campus, we have been using free-living organisms as a way to try to identify novel anthelmintics. And so we use our favorite organism, C. elegans, which has a very rapid life cycle. It's optically transparent. It has a huge molecular toolbox to get at mechanisms. And importantly, it's sensitive to most known anthelmintic drugs which are on the market. So it's sensitive to them. And then we also run our screens in another species, which is another free-living species related to C. elegans, but it's actually thought to be more than 100 million years divergent. And so this allows us to set up a system where we are trying to identify anthelmintics that would work across a broad range of different parasitic species. And so our workflow here is that we start with these screens. We start with screens of, it's hard to see from this location. So we have a multi-well plates that we put different molecules, small molecules in, and then we look for ones that can kill worms at some different concentrations. And the ones that are very good at killing the worms, then we take those and counter-screen them. That's the problem. Counter-screen them in mammalian cells to make sure that they might not, to find out whether they will be toxic or not to human cells, and therefore you wouldn't want to feed them to a host because it would make them very ill. And so the ones that pass this filter, we then go ahead and test in both parasitic nematodes in collaboration with people who actually have those systems in their lab. We also can screen multi-drug resistant animals, and that's really important because what we're looking for is novel mechanisms. And so if the molecules we find work in these strains which are resistant to various different anthelmintics, then we think, oh, that's great, we have potentially a new mechanism, and that would be really helpful in the field. So anything that we think might be a novel mechanism, we then can perform preclinical trials. And we don't actually shove big pills down mice's throats. We feed them in liquid form. And then we can eventually try to optimize the molecular candidates or go back to C. elegans and try to figure out what the molecular mechanism of action is. And so what we've done is we have quite a variety of different kinds of libraries. We also can screen genomic libraries using RNAi in worms, and we're also bioprospecting a little bit in the region to try to identify novel microbes that might have anthelmintic properties. So I'm going to tell you, and to do this, we set up this fancy system where we have a high-throughput robotic platform that I'm going to try to play the movie for a second for you with a moving arm, and we have a lot of different pieces of equipment, liquid handlers, spectrophotometers, and a high-throughput microscope that allow us, and incubators that are part of the system so that we can very efficiently screen a very large number of compounds or RNAi screens in a short time. And this is important for us because in New York, we had a lot of people in the lab spending really a lot of time and effort to do RNAi screens, and this has really been very enabling for us. So what could we do with this system? We use it to look at, we can measure OD600, and so we can see if the bacteria that we're, for an RNAi screen that we're feeding the animals is consumed, and if they are toxic to the worms, then we see a different result than if these RNAi do nothing to the worms, right? So then everybody's very happy. We can also measure paralysis, and we can do this high content analysis where we look at a variety of phenotypes in worms, and again, we counter screen in 1 million cells. So the first screen that I'm going to tell you about very quickly, I'll tell you about 3 screens, is this Cambridge library that we thought may be enriched for biologically active molecules in worms because it was, these were identified as very high priority in a machine learning approach in Peter Roy's lab as molecules that have increased bioretention in worms. And so we took 32,000 of the top hits from this, these predicted molecules, and we then screened them, and what we ended up with was 30 candidates, some of which were very potent, and we found some that actually kill Homunculus contortus and a couple that are able to kill Brugia pahangi, and so this was very encouraging. In fact, it was so efficient that we ended up having a problem of, I'm having a problem. Okay, we ended up with a situation where we actually had a very, very high hit rate. So we found 4,000 molecules that in our primary screen that we wanted to put through further screening. And this is really a lot for us. This just shows the HPLC assay that Peter Roy's lab did to test for bioretention. And then what they did was they took all the molecules that were retained, and then they tried to figure out what kinds of chemical groups were enriched in the ones which could kill the worms, which were accumulating in the worms, and so we what we did, because we had so many hits, is that we took the 4,000 hits that we had and we clustered them into groups of diverse molecular structures. And so we then were able to do a secondary screening on a little bit of a smaller set using one representative from each one of these groups. And we could later go back and see if any ones in the particular groups are more potent than the ones we selected. The second screen was a screen of drugs, chemicals which had previously found to have some activity in animals, or they're from this library that have that we got from the Caliber. It's the Caliber library. It's called ReframeDB. It was very hard to get because you have to put in a proposal, and we did this with our collaborator, Tony Page. And this is 12,000 compounds, I think, that passed. I'm really having some issues with this thing. That passed clinical trials. So they're not toxic to humans, but they weren't effective for their intended purpose. So we have access to these, and we screened all of those. So altogether, these two libraries, it was over 13,000. We again found a bunch of new bioactive molecules. Some were active in Brugia pahangi, which is a very nasty parasite. And then we did a third screen on this library called Spectrum Collection. And this is we got something very nice out of this screen, and I'm going to then just talk about that for the rest of the talk. So what we found in this screen was that this is a screen on C. elegans and Pristioncus, as I told you. And what we found was that we need a drum roll or something. That what we found is that although we didn't always have an effect of these are the dark gray or known anthelmintics that we tested, and we found that we could detect a phenotype in both C. elegans and Pristioncus for most of them. Pristioncus was more sensitive, though, because sometimes we could see an effect in Pristioncus and not in C. elegans, and we could see a more severe phenotype in Pristioncus than C. elegans. So that was kind of interesting. We also found a nice molecule, which is Zoloft, and we were kind of excited about that. We found an antidepressant that can be repurposed as an anthelmintic. So if you're taking Zoloft, you might be, have less, propensity to get worms infection, like Kennedy. You might not get a worm in the brain. But this unfortunately was published already, so other people beat us to the punch with that one. However, we found this group of 6 related molecules that are 17-carbon alcohols in their acetate forms that are derived actually from avocados. And so that was something we were very curious about. And so we chose to follow up on these first, also because they had pretty strong phenotypes and a pretty low LD50. So this just shows the structure of these related molecules. There are some combinations that are already known. They're called avocatin A and B, but we mostly tested avocadine acetate because it was one of the most potent isoforms. And then we also tested these 2 furans, which are in the library, and found that they had no effect. So there's something to do with these alcohol and acetate groups that was important for the function. So we tested them, found they didn't have an effect on mammalian cells, but they can kill all different stages of C. elegans. And we also were able, because we have this NMR expert in the lab, Rino Esposito, he was able to make C13 labeled extracts and demonstrate that he could detect a very specific signal for these molecules, both in larvae and in embryos. So that was very, very encouraging to us. We also found that, interestingly, whereas most of these anthelmintics, as I mentioned to you, can kill adults, they do not effectively kill, for the most part, except for praziquantel in Pristioncus, they do not kill the eggs. And this is important, whereas our molecules, what we're calling, this one is avocadine acetate, but we can call them avo molecules informally in the lab, and we published a paper on this, and we call them avocado-derived fatty acids and alcohols, which is a bigger mouthful, but I could abbreviate as AFA. So that's the other thing you might hear me say. And this is important because, as I told you, the animals shed eggs into the environment, and so something that actually can kill eggs is very, very useful. We also investigated the phenotypes, specific phenotypes in C. elegans, and we found that in embryos, within 20 minutes, they will arrest development. So if you compare the control animals, which have many, many cells at this time point, these are stuck at the 4-cell stage. We also found that they cause paralysis. They also inhibit pharyngeal pumping. And they also will use this machine called the seahorse to measure mitochondrial respiration. And we found that they inhibit mitochondrial respiration. And under severe conditions, we also found really weird mitochondrial phenotypes where the mitochondria kind of round up, and then sometimes they even just totally disappear. So it's quite toxic to worms, and somehow humans do something else with these molecules. So that's pretty cool. So eat a lot of avocados, apparently. We also, with our collaborators, tested against in L2 stages of a bunch of different parasitic nematodes that infect a variety of different organisms. These are the models that you can test. And importantly, we found that even in multi-drug resistance Homunculus contortus isolated from the field, and in C. elegans mutants that have genetic mutations that make them resistant to different drugs, they worked in all of these drug-resistant animals. So that was super promising. And this just shows that in these different mutants, which are resistant to different molecules, to different known anthelmintics, the LD50 was pretty much in the same ballpark in all of these different strains. Then we, together with our collaborators at the University of Glasgow, we pre-infected mice for 9 days with H. polygyrus, and then we fed them avocado-derived molecules by lavage. Lavage would be cleaning them. No, this is gavage, which is the way they make pâté. And what we found is that after about 3 weeks, we had a great reduction. Sorry, I'm having technical issues today. We had a really great reduction in both the number of eggs that we were shed into the feces, as well as the load of adults that remained in the animals. So these were very encouraging results. So, oh my. Well, you just saw the whole talk. I guess we're done. Let's see, where were we? We were getting there, don't worry. I can't seem to go back. So it's okay. I got it. It's okay. No, I don't. Why is it not going back? Okay, I'm going to do this very quickly. So close your eyes for a second, and then we'll... Okay, this is fine. So, what? Oh, to go back. Okay, that's fine. What is going on? Okay, it's not just me. Something very strange. Pardon us while we undergo repairs. Oh, it's this thing. Well, I pressed it because nothing was happening. I should just wait. I should just be patient. What's that? This one? Yes. Okay. Oh, okay. Yeah. That's fine. Yes, this will make my life easier. The laser point. Okay. So where was I? As I was saying, one way to discover a mechanism of action is to find a genetic mutant in an EMS screen. So ethyl methane sulfonate, it generates point mutations in the genome. And sometimes you can do an EMS screen and then find really important genes and win a Nobel Prize. But that didn't work for us because even though we tested a large number, almost 10 million genomes, we were unable to find a single resistant mutant to these drugs. So that's not so great for us to try to find mechanism, but it's fantastic if you want to get a new drug that can kill parasites if you can't generate resistance to it. So instead we turned to molecular profiling and genetics, and we did some transcriptomics and metabolomics using our fantastic, core resources, but also we sent some stuff to a company, which now we can do it here. I think it's much better because if you want to go back to the company and say, what about this one molecule you didn't profile? They'll say, we can look for that, but give us another $10,000. And we didn't want to do that. But anyway, so what we found is that in the metabolomics data, that there was an increase in acetyl-CoA accumulation after treatment with these avocado-derived compounds. And we thought, okay, that's a good lead. And we then found a mutation. There's a gene that is one of the largest consumers of acetyl-CoA in the cell, and it's called, more generically, acetyl-CoA carboxylase. And it's the first committed step in fatty acid biosynthesis. And so we had an NCL against this gene is called POD2. So we had a POD2 null mutant, and what we found is that they were somewhat more sensitive than the wild type. And in both cases, if we gave them the product of this enzyme's reaction, which is malonyl-CoA, then we could rescue, to some extent, the lethality. So what is this enzyme? ACC is a multi-subunit, it's a multi-subunit enzyme. It operates as a dimer. And the most important thing here is just that it transfers a carboxyl group to acetyl-CoA, and that generates malonyl-CoA, and then you can continue building the chain, and then you end up with C16 that you can make desaturate. You can desaturate it, and you can add other things, and then you get membranes, which is really, really important for development. And so this reaction here of this transfer occurs in this CT domain here, this red thing. And it's interesting because you need the dimer for the reaction to go because the active site moves to the subunit in the other half of the dimer. So they're completely inactive as monomers. Okay, so this is the basic scenario. And what we did a bunch of molecular modeling because we have a wonderful structure guy in the lab who does this. And when we looked at, modeled the POD2 dimer, and we looked at how this avocado molecules might bind to this protein, we had a very, very nice prediction that these AFAs would bind right in the CT active site in such a way that it should block the ligation of CoA to biotin, which is actually the first step in the reaction. So this looked like a really, really good candidate. So what we did was we then took advantage of having Reno in the lab, and he was able to detect a difference in an NMR spectrum between these acetyl-CoA and the malonyl-CoA basically by a shift in the ppm. And so you can see if you combine your, it was actually a partially purified extract from worms. If you treat them with avocadine and then look after a while, you can see that you have a great reduction in acetyl-CoA and an increase in malonyl-CoA. But he developed this method where he could monitor essentially in real time the reaction, so you could see the depletion of acetyl-CoA at the same time that you get an increase in malonyl-CoA, which is super cool. And here is a typical time course that he found. And you can see that acetyl-CoA is consumed and malonyl-CoA is produced. This, unfortunately, we have a little bit of contamination with some other enzymes that also use biotin. And these have, on the other hand, a different product. So they don't produce malonyl-CoA, they produce CoA, so we can easily distinguish our ACC activity from the activity of the contaminating enzymes. And when we we can also compare the accumulation of malonyl-CoA when we, in the absence of these molecules or with an increasing amount, and we can see that we get an increasing inhibition of this enzyme. So that seemed like we had it nailed, although I have to point out that because we have a lot of different phenotypes, there may be other targets. For example, this is probably not responsible for the paralysis that we see in the worms. There's probably an additional effect on the nervous system or musculature or something. And we're actually, Fabio has a postdoc who's looking into this, right, Hannah, right now. Okay, so I told you that they're much better at killing eggs, so you get much less egg hatching with these AFAs than with other known anthelminthics. But the interesting thing is we also had Yamanapa in the lab who was able to use click chemistry to attach a fluorescent dye to the end of these molecules because they have the alkynes had a triple bond at the end, and that made them very amenable to ligating some molecules. And what we could see here is then with this fluorescent molecule, we can actually see it accumulating in embryos when we treat the embryos directly. So that means it's getting in through the eggshell, which is very, very unusual for any molecule to get through the eggshell and C. elegans. But because they're sort of lipid-like, it may help them get through the eggshell and the waxy barrier that surrounds the embryo. And so we also found two other known inhibitors of ACC, but we found that they didn't work unless you crack the eggshell. So this is just AFA, and then these other two also cause a developmental arrest, but they only work if you smush the embryos a little bit so that they can penetrate. And I think that I'm going to be beating a dead horse by telling you this is really important because these shed eggs into the environment. So we were very excited about this result, and we managed to publish this in a nice publication. So you can read all the details, which are not at the top of my head. And you can also, so just to summarize, we established this pipeline. We screened a lot of many, many thousands of chemicals. We identified a variety of new chemical classes that seem to operate by different mechanisms than the known anthelminthics. And they seem to work also in the field, both upon direct application and in an animal model. And we identified a target of these molecules. So Fabio wanted me to show you that he loves ChatGPT, and ChatGPT loves us because he asked it to make a 1-minute podcast for young people about our paper, and I'm just going to play it for you very quickly.
Today we're exploring something amazing. Avocados might be our secret weapon in fighting harmful parasitic worms that make people and animals sick and damage our crops. Scientists have uncovered an incredible discovery hiding in avocado seeds. Inside these seeds are special natural chemicals called AFAs. When these chemicals meet the parasitic worms, they disrupt the worms' energy systems. This stops the worms from moving around, growing bigger, and even prevents their eggs from hatching. It's like turning off their power switch. The best news is that these chemicals are completely safe for humans and animals. In tests with mice who had these parasites, the results were incredible. After just a few weeks of treatment with avocado seed chemicals, almost all the worms were gone. This discovery means we can turn avocado seeds, which usually end up in the trash, into powerful medicine that helps people, animals, and our planet. So remember, when you're enjoying your next bowl of guacamole, those avocados are more than just tasty. They're part of an amazing scientific breakthrough that could help protect our world.
I have never heard this podcast before. So, super cool, right? All right. So finally, I just want to thank the people who were involved. This is our lab in we have a lab in Abu Dhabi and in New York. This is a picture of the campus before all the stuff that got built around it. So at the beginning, right after it was constructed. And so it was really in the middle of nowhere for a while. This is just a picture of New York. That's pretty. I want to thank in particular NYU Abu Dhabi for funding the lab, for the funding the CGSB grant, the NYUAD Research Institute, and also the Ziegler Family Foundation, who has provided some support. These are the people in the lab, mostly in Abu Dhabi, but a couple people helped in New York. I want to particularly thank Hala for guiding and leading this project the whole time. This is the experimental group. Nabil, Yanthi, and Glenn have helped us with the computational work. Reno and his team have done the biochemistry and the NMR. And then we also had a couple capstone students involved in this project. And one of them went to Glasgow and screened a bunch of new molecules against the parasitic models that our friends have. And so this is Hala. So if you see her and you want to talk more about the paper, you can find her at the breaks or tomorrow. This is Hala and Fabio at a meeting. I also would like to thank our collaborators, Tony Page, Rick Maisels, and someone in their lab, Claire, who have done all of the animal work, as well as the core technology platforms that support our research here. They're totally amazing, and we're very lucky to have these resources here, and Wormbase and the CGC for strains and information. So just so you know that Hala and Fabio don't just hang out at meetings with cats, they also are always working, and this is them on the way to that same meeting in the airport, going over all of our data. So this is the paper that we just came out, and if you're interested to read more about it, that's a link to the paper. And I'm happy to take questions. Thank you for your attention.